Welcome back. In this section, we're going to learn all about transcription, that process of going from DNA to RNA. So let's get started. This process of transcription has 3 steps. Step 1 is known as initiation. Okay. So to understand initiation, the first thing we have to know is that DNA is composed of many genes. And at the beginning of every gene, there is a start signal. And this start signal is known as the promoter. And here in this picture, they've highlighted as yellow. Okay. And in order for initiation to occur, there is an enzyme known as RNA polymerase that is going to bind to the promoter. Why would we be using RNA polymerase? That's right, because we want to make RNA this time, not DNA. Okay. So RNA polymerase is the enzyme that's going to be attaching to the DNA at the promoter site. And so now we're going to be opening this double helix. We're going to be forming a bubble just like we did with the DNA replication. In this case, only one strand is going to act as a template because remember, RNA is single-stranded, so we're only making one strand of RNA. Now, the second step of transcription is called elongation. So in elongation, the process of transcription is actually occurring. We're going from DNA to RNA. In other words, we're going to be attaching complementary bases. So in the DNA, we have C, G, A, and T. And so now we're going to be using the complementary bases of C, G, A, and U. So the pink bases are the ones that are being added. So you can see wherever there is an A in the DNA, we are going to be adding the U for RNA. And don't forget the enzyme is RNA polymerase because we are making RNA. RNA bases, RNA nucleotides are attaching to the DNA template. Okay. And this is going to continue all the way down that bubble. The RNA polymerase is going to continue all the way from the promoter until it reaches a terminator, also known as a stop sequence. And that will bring us to step number 3 of transcription, known as termination. Okay, so when it reaches that terminator stop sequence, the RNA is going to detach, the RNA polymerase is going to detach, and the original hydrogen bonds of the 2 strands of the DNA are going to reattach, and so the double helix is going to reform of the original DNA. It is not harmed in the making of the complementary RNA strand. So remember, only one complementary RNA strand is made. It's made to the side that had the promoter and the terminator gene on it. Okay, so now we have a strand of RNA. We do not have a strand of messenger RNA. We just have a strand of RNA. So what's included in this strand of RNA? We've got a lot of stuff going on. And if you look at this, you can see that they've used a couple of different colors. We have blue colors and yellow colors. Okay, and we have 2 different areas here. We have areas known as exons, which are areas of RNA which will code for stuff. And we have areas known as introns, which don't code for anything. They're nonsense areas. So we actually are going to have to remove the intron areas. Okay. So introns are actually nucleic acid areas, but they're not going to code for amino acids. So after that RNA is formed, we have to do a little nip tucking, a little plastic surgery on it. Okay. And we want to make sure we leave the exons, but we can't just have little strips of exons floating around. Once we remove the introns, we've got to reattach all the exons. So we got to figure out how to put them back together. Okay. So this process of editing is called RNA splicing. And in this splicing is how we're going to be taking this strand of raw RNA and forming our messenger RNA, which will be exiting the nucleus. So we're going to start with this raw piece of RNA, which has exons and introns. It also has extra nucleotides that form a cap or beginning at the 5' end and a tail or an ending at the 3' end. Why is that? In case there's damage, it's not going to be ruining any coding regions. Okay. Then the introns are all removed. Okay. All the yellow areas are cut out, and then all the exons are left behind and spliced together. Notice the cap and the tail are still left there. And this is your messenger RNA with the cap and the tail that's going to be exiting the nucleus. This edited piece of RNA is now called messenger RNA. Okay. And it exits. Okay, so with this, I would like you to review this process of transcription. On the left, name all the steps and name these areas and name this purple enzyme and where did this occur, and then go over to the right side and I put about 6 different questions there for you. And that's it for this area. So thank you for all your work, and I will see you when we come back and we talk about translation. Thank you.